Hi students, welcome to Ligis Academy. In our previous video, we are already discussed about different stages in transcription. As a result of transcription, messenger RNAs are formed. Then what will be the fate of that mRNA? And what are the processes involved in the transcription of transfer RNA and ribosomal RNA. In this video, we are discussing about fate of mRNA and the transcription of tRNA and rRNA. So, we are discussing about fate of mRNA. What happens to mRNA after transcription? As we all know that in prokaryote, transcription and translation are a coupled process. That is, transcription and translation occur simultaneously. Prokaryotic mRNA have very short half-life, that is approximately 1 to 2 minutes. So it is degraded very rapidly from 5' end. You just imagine that if it has high half-life, all previously synthesized mRNA will compete with an nascent mRNA for ribosome. So, avoiding this competition, it has very short half-life. In messenger RNA, there is a triphosphate at 5' end. It is the only component that increases the stability of mRNA. So, it greatly influences the mRNA being translated by ribosome. So, ribosome has a function in protection of mRNA from degradation. To rescue from the degradation, this mRNA translated before being finally transcribed. So, the ribosome has role in protection of mRNA from degradation. As we already mentioned that prokaryotic mRNAs have very short half-life. So, it degraded immediately after the transcription. The degradation of mRNA with the help of many enzymes. Several enzymes involved in the degradation of mRNA. This group of enzymes are known as mRNA degradosome. mRNA degradosome is a multi-protein complex. Then coming to the steps involved in the degradation of mRNA. As we already discussed that the stability of the mRNA increased by the presence of triphosphate at its 5' end. So, removal of this triphosphate from 5' end will reduce the stability of mRNA. So, the first step in the degradation of mRNA involves this removal of triphosphate. The enzyme RNA pyrophosphorhydrolase or RPPH removes this terminal pyrophosphate from 5' of mRNA. The picture represents the removal of pyrophosphate from the mRNA with the help of the enzyme RNA pyrophosphorhydrolase. Next, the degradation of mRNA with the help of enzymes RNase E. RNase E is an endonuclease that involved in the primary endoribonuclease cleavage. This RNase E cleave the mRNA after every adenine uracil dinucleotide. So, with the help of this RNase E, mRNA cleaved into fragments. These fragments of mRNA are degraded to mononucleotide by degradosome. Degradosome is a multi-protein complex. Then the polynucleotide phosphorylase or PNP degraded this 5' fragment into mononucleotide. This polynucleotide phosphorylase is a 3' 5' exonuclease. So it can act on the 3' fragment because there is a presence of stem loop in the 3' fragments. So for degrading the 3' fragments, the enzyme poly-A polymerase acts on it. This enzyme poly-A polymerase add poly-A tail in 3' end. The action of this poly-A polymerase using ATP as a substrate add poly-A tail into the 3' termini of both full-length mRNAs and the degradation product that contains secondary structures. So presence of poly-A tail serves to facilitate the degradation of mRNA molecule. So addition of poly-A tail making it susceptible for polynucleotide phosphorylase. So the polynucleotide phosphorylase degraded this fragment into mononucleotides. This is the schematic representation of degradation of mRNA. The mRNA degradation involves endonucleolytic cleavage as well as exoribonucleolytic cleavage. First, the endonucleolytic cleavage with the help of RNase enzyme, RNase 3 and RNase E. It cleaved this mRNA into fragments. After the fragmentation of this mRNA, exoribonucleolytic cleavage occurs. That is, polynucleotide phosphorylase degraded this fragment into mononucleotide. In the absence of RNA endonucleolytic cleavage, the polyadenylation dependent pathway is promoted. In this case, polyadenylation phosphorylase add poly-A tail in the fragment and it will facilitate the action of exoribonucleolytic cleavage. Then finally it will make mononucleotide. Then we are going to discuss about transcription of transfer RNAs and ribosomal RNAs. Transfer RNAs, otherwise known as transport RNAs, are responsible for transporting amino acid to ribosome for protein synthesis. Then ribosomal RNAs are associated with the ribosomal protein to form ribosome. The primary transcript of both transfer RNAs and ribosomal RNAs undergo a series of processing steps. In prokaryote, 3 ribosomal RNAs exist: 23S, 16S and 5S. These tRNAs and rRNAs are coded by 7 operons. They are rRNA, rRNB, rRNC, rRNE, rRNF, rRNG, rRNH. These operons code for all types of ribosomal RNAs, but each operon specific for particular type of transfer RNAs. The order of rRNA in all operons are same, that is 16S, then followed by 23S, then 5S. There are spacer region between 16S and 23S and after 5S. These spacer region code for the tRNA. There are 2 promoters that contain upstream element and Fis site. The primary transcript will contain both tRNA and rRNA. There are 2 promoters P1 and P2 in this operon that contain upstream element and Fis site. When the transcription of this operon occurs, the primary transcript form. This primary transcript contains both tRNA and rRNA. So, it later processed into ribosomal RNAs and transfer RNAs. So, next we are discussing about the processing of rRNAs and tRNAs. Let's talk about rRNA processing. The primary transcript undergoes initial cleavage. The initial cleavage of this bacterial pre-rRNA yields a separate precursor for 3 individual rRNAs. This is known as pre-rRNA. These pre-rRNA are further processed by secondary cleavages to the final product. There are specific cleavage sites for RNase 3 that make a symmetrical cut in the stem region of 16S and 23S. This picture represents the processing of rRNA. The primary transcript consists of both rRNAs and tRNAs. With the help of RNase 3, it releases pre-16S and pre-23S rRNAs. This pre-16S and pre-23S rRNAs then further undergoes modification to produce mature rRNAs. The modifications in the bases of these pre-rRNAs are important for the maturation of rRNA. This is the complete representation of rRNA processing. The primary transcript consists of both rRNA and tRNA. With the help of enzyme RNase 3, it cleaved into pre-16S, pre-23S and pre-5S rRNA. Then it undergoes modifications and forms matured 16S, matured 23S, matured 5S rRNA. And this maturation involves the modification of bases. Modification of bases include methylation, pseudouridination and O-methylation. Then talk about the processing of transfer RNA. Transfer RNA has important role in protein synthesis. Like rRNAs, tRNAs are synthesized as longer precursor molecules. It is known as pre-tRNAs. In bacteria, some tRNAs are included in the pre-rRNA transcript. The processing of the 5' end of pre-tRNA involves cleavage by an enzyme called RNase P. These RNase P cleave the 5' end of pre-tRNA because this RNase P is consists of RNA and protein molecule. It itself capable of pre-tRNA cleavage because this RNase P is a ribosome. That means the enzyme in which RNA rather than protein responsible for catalytic activity. So this RNase P cleave the pre-tRNA at the 5' end. All tRNAs have CCA sequence at their 3' end. This sequence is the site of amino acid attachment. So this CCA sequence is required for tRNA function during protein synthesis. So it encoded in the DNA of some tRNA genes, but in others it is absent. If this CCA terminus encoded genes are absent, in that cases CCA terminus being added as an RNA processing step. This adding of CCA terminus by an enzyme that recognized and added this sequence. The bases in tRNA molecules undergoes modifications. That is, approximately 10% of bases in tRNAs are altered to yield a variety of modified nucleotide at specific positions in the tRNA molecules. This modification of bases have important role in protein synthesis by altering the base pairing properties of tRNA molecules. These are the steps involved in tRNA processing. The tRNA primary transcript consists of 2 stem loops. Stem loop 1 at the 3' end and stem loop 2 at the 5' end. An endoribonuclease recognize stem loop 1 and cleave to generate 3' end. Then exoribonuclease degraded 7 bases from this generated 3' OH. This process slows down when it reaches the UC dinucleotides. An endoribonuclease RNase P that recognize stem loop 2 on the 5' end and make a nick to generate 5' phosphate. RNase D also degrade remaining dinucleotides. The 3' end of tRNAs is generated by the action of conventional protein RNAs. But the processing of this end of the tRNA molecules also involves an unusual activity, that is the addition of CSIR terminus at the 3' end. All tRNA molecules 3' end terminate at a CSIR sequence. If there is no CSIR end, it will be added by CSIR adding enzyme. Because this sequence is the site of amino acid attachment, so it is required for tRNA function during protein synthesis. Then the next processing, the loop region in the tRNA are modified to generate mature RNA. Mature tRNA. That is, the modifications have important role in protein synthesis by altering the base pairing properties of tRNA molecule. That's all about this section. Hope you all understood about the processing of rRNA, tRNA as well as the fate of mRNA. Stay tuned to next video on eukaryotic transcription. If you like this video, please subscribe, like and share. Like is Academy, your educational catalyst.